Okay, the live stream has started on YouTube as well.
Okay.
Okay, I think a few people have joined in, so
All right.
let's begin. Yeah, people can keep joining in as they come. So yeah, I'll start with a quick introduction and then I'll let you take over.
All right. Thank you.
Okay, so yeah, Dr. Catarina de Almeida Marques, or Cat, as she would like to be called, is a postdoc at the University of Glasgow in UK. Her research focuses on oops, that was hard. I lost my webcam. Just give me a second to reposition that.
Okay.
Yeah, I think that should be fine now. Yeah. So her research focuses on how DNA replication takes place in Trypanosoma and Leishmania, which are two protozoan parasites that infect people and cause various diseases in many parts of the world. So she started her undergraduate degree in microbiology at the University of Lisbon in Portugal, and since then she has been interested in bacteria and viruses. And now her curiosity has shifted towards protozoan parasites, and particularly during her final year internship, where she was studying about Plasmodium, which causes malaria. So that's quite a diverse background in microbiology, Dr. Okat. So I'll let you take it. I won't take too much of your time now.
No, it's okay. Thank you very much. I hope it all works and the transcript actually translates what I'm actually saying. First of all, I would like to thank you guys very much for the opportunity to talk, and also for this amazing gathering and conference. I think it's a great idea. And also for the server, which I find great. So thank you very much for all of that. So today I'll be talking to you like hello, good afternoon, morning, evening, depending where you're streaming from. I'll be talking about a couple of topical parasites and the odd case of how they replicate their genomes. So as Anchit said, I'm Catarina, and I'm a microbiologist, and I am, this is not working. I am originally from Portugal, and it was in Lisbon that I did my undergrad and master's in microbiology. And as he said, I started to drift a bit into parasites, although that was not my initial intention. But in my final year of undergrads and then during my master's, I shifted and I started focusing on the malaria parasite, which is a tiny little guy there. And then I haven't left parasites ever since. So I am a microbiologist, but for a while now, I've been mainly a parasitologist. So after my master's, I decided to do a PhD, and I decided to do it somewhere else for several reasons. One of them being having experience somewhere else. So I moved to Scotland, more specifically to Glasgow, where I did my PhD. So that's where I started to study DNA replication. And that was in trypanosomes and in Leishmania. And after that, I moved a bit up north to Dundee to do a postdoc in trypanosomes, where I studied the cell cycle and also one of the factors that are involved in the expression of the surface coat. And then I got back to Glasgow, to the same place where I did my PhD, to follow up some lines of research that stemmed from my project, and just to try and dig a bit deeper into how these parasites replicate their genome. So my talk merges two fields, so parasitology and molecular biology, namely DNA replication. So I know that the audience is quite dynamic and diverse, so people at different stages of education, from undergrad or even high school to postdoc, etc. So I decided to make more of an introduction as well, so that everyone is on the same page and everyone can actually grasp a bit of what I have to say, and I hope everyone enjoys it. So I'll start first to introduce what these parasites are. What are these parasites I'm going to be talking about? So I'm going to be focusing on two, so the trypanosome and the leishmania. So the trypanosome I'm going to be talking about, and there are several species within that genre. I'm going to be talking mainly about the African trypanosome, or the Trypanosoma brucei and its subspecies. So T. brucei is the pathogen that causes sleeping sickness in humans or nagana in cattle, and it is endemic in sub-Saharan Africa, and it affects a lot of countries in a wide geographical area. So in 2019, there was only reported less than 1,000 cases, although it's believed that there are a bit more and not all of them are reported. But because it affects such a vast area, around 65 million people are actually at risk every year to get it. And this disease is really severe. If you don't get treated, it's fatal. And even when you do get treated, it depends on the stage that has been diagnosed, and the prognostic is quite reserved. So the other thing is that the drugs are rather nasty. They're not nice, and they're pretty old. When I say old, it was like they were developed a long time ago, but they're the best that we have. So there's a lot of research to try and develop new drugs and find new drug targets for this disease. So the parasite is restricted to this area because it's here where its vector actually lives.
Hi, Kat.
Sorry?
Are you sharing your slides?
Sorry.
Like, are you on your presentation? We can't see the slides moving.
Yeah, I'm still on my presentation. Sorry. Oh, it says your shared window up to the front. Okay, what does that mean?
I can only see your first slide. I'm not sure if all the attendees are facing the same problem.
So I'll stop share. Stop share. Sorry. I must have clicked on something else. Okay. Wait, give me a second. So where did you lose me?
I think it was the third or fourth slide.
Sorry,
yeah.
where?
Yeah, this one.
Here.
Yeah.
Okay. Is it popping in? Yeah.
Yeah, it's we can't. Are you in your presentation mode? I can see it zoomed out, but I can see the slide now.
Okay, does it move?
It's not moving. I'm not sure why that is happening.
Yeah, it says sharing is pausing. Bring your share window to the front. Resume sharing. Okay. Right. I'll take the subtitles off.
Okay. That might be causing the issue.
Can you see it now?
No.
No. Yeah. Sorry about that.
That's all right.
That's fine. Not really sure what's going on. Take the subtitle. I don't think it's subtitles, but so if I put presentation mode, it stops sharing.
Yeah, and the presentation was stopping to share.
Okay. Sorry. You need to click back on the presentation view. Okay, someone says that they know how to solve that problem.
Oh yeah, that's Dr. Frog's one of the moderators.
Yeah.
We'll just move her up to a panelist.
Okay, so.
Yeah, Dr. Frog should be here.
Hi.
Hiya.
So if you do share screen and you click advanced options, there's an option to share only part of your screen. So when you do, if you share presenter mode, then you can adjust your window to just share the slides as opposed to the notes as well.
So if I click on share screen, yeah, I just select the PowerPoint one.
So what I do is go to advanced options when you click share screen.
Oh, right.
and then it says share only part of your screen.
Portion of the screen, yeah.
That's it, yeah.
Okay.
And
Okay.
then you adjust the window to just fit your slides.
Oh,
That should work.
okay. That never happened before. Okay. Can you see it now then?
Yeah. So if you tried changing now, if you tried changing the slides,
Yeah.
yep, that's working perfectly. Great.
Okay. Right. Do you want me to continue where I stopped, or do you want me to go back?
I think we can start one slide before this one.
Okay. Yeah. Oh, right. Right.
Yeah, that should be fine.
Okay. Yeah. So, yeah. So no one saw the introduction bit. Right. Anyway. Right. Yeah. So as I was saying, it's two parts. It's two areas. So it's parasites and then DNA replication in eukaryotes. So as I was saying, I'm going to introduce first what the parasites are, and then what DNA replication is. Well, what we know about DNA replication in eukaryotes as a general knowledge. So I had started with the parasite one. I apologize because it was definitely passing for me, the different slides. So I didn't realize that it's not actually doing anything. Yeah, so I was talking about Trypanosoma brucei, and so it causes sleeping sickness. And in humans, and it causes another disease called Nagana in cattle, which leads to cattle waste. So it has a huge economical burden on the veterinary aspect of it. It is endemic in sub-Saharan Africa. And although less than 1,000 cases were reported in 2019, it's thought that that's under-reporting. However, due to the large distribution of where the disease is endemic, that means that around 65 million of people are at risk every year of getting it. And if left untreated, it is fatal. And even the treatment is quite harsh, and there's resistance issues, etc. So there's a lot of effort to try and develop new drugs against these parasites. The parasite is restricted to this geographical area because this is where its vector lives. So the trypanosome is a vector-borne disease, and it's transmitted by the tsetse fly, which only lives in these areas. So it has two hosts, the fly and the mammalian. And it oscillates between several morphological forms. So it changes how it looks and it changes how it acts depending on the environment that it's in. So in the fly, it goes from an infective stage, which is called metacyclic, that then is injected into the mammalian host. And then just before it develops into that stage, it goes through several rounds of replication as the promastygote and then other differentiation steps. Once in the bloodstream of the mammalian host, it changes into a bloodstream form that then replicates, and that's what causes the disease symptoms. And then eventually differentiates into a transmission form called the stumpy form. And that's what's eaten by the fly and then starts it all over again. One characteristic of trypanosome, the African trypanosome or Trypanosoma brucei, is that it does something called the antigenic variation. So it expresses on the surface, so it coats its whole surface with a protein. But because they are exclusively extracellular, they're constantly exposed to the immune system, right? So the immune system develops antibodies against, for instance, the red coats. What happens is that in the population of parasites, there's going to be some that have a different color of coat. So let's say yellow. So when the immune system destroys the reds, the yellow take over the population. And then again, the immune system develops antibodies against yellow, then purple takes over, etc. So you actually have waves of infection, and that's very characteristic of this parasite. The other parasite that I'm going to be talking about is Leishmania. And Leishmania causes cutaneous and visceral disease and also infects both humans and animals. So it's a huge problem of human health, but also veterinary health. There's around 1 million cases a year, and because it's distributed all over the globe, actually around 1 billion people are at risk of getting it. There's lots of Leishmania species. That's why I'm not going to focus on a single one for this part. And in some countries, the human infection is the main concern. In other countries, it might be the veterinary. And that's also an issue, not just for the veterinary point of view, but also because it acts as a reservoir. Just like the trypanosomes, is also a vector-borne disease, but this time is transmitted by the sandfly, which exists in many places. And while in the fly, it is a free form, well, it is extracellular parasite. In the mammalian, it infects macrophages and changes dramatically in shape and grows inside the macrophages. Both of these parasites are eukaryotes, and here is just a very simplified diagram of the eukaryotic tree as it stands at the moment. So just to put it a bit into context, we humans are here in the metazoans, and together with fungi, we are in this other group called the Opisthocomta. And I just call attention to this because this is where you have normal yeast, you have C. elegans, which is a worm that is used as a lab model a lot. We have the fruit fly, the mouse, the zebrafish, and all these are considered model eukaryotes. And when I mention model eukaryotes, I mean it's organisms where lots of research has been done for various reasons. One of them being that it's close to humans to a certain extent. So research in there can then be translated easily into human health. And what has happened over the years is that most of the technologies and new techniques and knowledge has been developed in these organisms. So lots of studies are just done in this as a continuation of that. One thing that has happened until very frequently and sometimes still happens is in studies done in these organisms, sometimes it's said that the findings are conserved across the eukaryotic tree because they can find it between yeast and humans. The problem with that statement is that, as you can see, the tree is pretty big and all of these are in the same small group. Just for you to have an idea, plants on these other sides, Plasmodium, who causes malaria, and Toxoplasma, which are also protozoans, are in this SAR group, in the same supergroup. And the parasites I'm going to be talking about today are actually in another group called the Discoba, which is a completely different branch of the eukaryotic tree, but whose relationship with the other branches is not very well established. And within the Discoba, Leishmania and trypanosome are inside a group called the kinetoplastids. And the kinetoplastids are some really odd eukaryotes when you think about it. So I'm just going to give you a brief introduction of what kinetoplastids are. First of all, it's a funny name. It's a weird name. Where does that come from? It comes from a cellular structure called the kinetoplast. So here is the nucleus, and here these two are the kinetoplasts. Kinetoplast is nothing more, nothing less than ultra-condensed mitochondrial DNA. And you can see it down the microscope, so it's quite impressive. So kinetoplastids all have a kinetoplast. The other feature of these organisms is that they have single copy organelles. So they only have one mitochondria, they only have one Golgi, etc. So, and their localization and structure is quite rigid. So that's also pretty odd and interesting. In the case of trypanosome, for instance, the mitochondria, it's quite elongated and goes along the whole body, which is pretty cool. They only do endocytosis and exocytosis from a single site in their body, which is the flagellar pocket right here where the flagella stems out. The part that interests me the most is that they have an odd genome organization. As you might know from like just high school biology classes, each gene is controlled by a promoter, right? That's what we're told. And that's how most of the eukaryotes that we know work. So a gene is expressed from a promoter. In kinetoplastids, what happens is there is one promoter for a huge group of genes. So one promoter transcribes dozens of genes, very similar to what you see in bacteria with the polycistrons. But in this case, the genes might not be related at all. So as a consequence, they have polycistronic transcription. So they generate this gigantic mRNA that needs to be then chopped off into individual messenger RNAs for each gene, right? They do that by transplicing, which is also pretty cool. And because of that odd transcription, all the control on protein levels and on products is all post-transcriptional. So it's not at the transcription level, it's then at the translational level and protein maintenance and degradation. They also have an odd DNA base, which is called base J, and it's hypermodified thymidine, which is also pretty interesting, and it's involved in transcription termination. Around 40% of the genes have a known function. That is quite a large portion of the genome, so there's a lot we don't know about how these parasites work. They have highly divergent proteins, pathways, and complexes that you either can't find in other eukaryotes, or the sequences are so different that it's hard to find parallels. And regarding the cell cycle, there's lots of checkpoints that are missing or that we haven't been able to identify because things are very divergent. So now that I tackled the parasites a bit, I'll just brush off what we know about DNA replication in eukaryotes. So here's just a simplified diagram of the cell cycle. So we have the GAP1 phase, the synthesis phase, GAP2, and mitosis. And as lots of you might know, DNA replication takes place in the S phase. So in normal cells, and I'm not talking about stem cells or germline cells, nothing like that, just normal autosomic cells, DNA replication takes place strictly in S phase. And this must happen once and only once during the cell cycle. And to ensure that this is the case, there's lots of control mechanisms in place, and some of them are even redundant. So in the case of one failing, the other one takes over. And this is very important to maintain genome stability. If you don't replicate everything in time, then one of the daughter cells is not going to have the entire genome content, and that will cause problems. If they replicate more than they should, so if they re-replicate parts of their genome, then you have extra copies of what you should have, and that will lead to problems. So for instance, one of the most many cases of cancer are caused by problems in cell cycle control and DNA replication. So DNA replication starts at specific sites in the genome, so it doesn't start randomly. It needs to be in a specific place, and these places are called origins of replication. So in eukaryotes, there are multiple origins per chromosome, and this is necessary to guarantee that everything is replicated by the end of S phase. The other thing is that there's always more origins than the ones that are needed in case the replication fork gets stuck, collapses, etc. It doesn't matter as long as everything is replicated by the end. So there's lots of safe mechanisms. An origin is defined as an origin in eukaryotes not by sequence, with the exception of yeast, where that is the case, but not in any other eukaryote studies so far. An origin is an origin because an initiator complex binds to it. And in this case, it's ORC, and I'm going to be talking about that in a second. After the initiator binds to the origin, it recruits a helicase that will open the DNA. And this helicase is recruited via a specific loader. The thing is, all of this happens before S phase starts. So this is just to get everything ready for when S phase starts. So at the G1 to S phase transition, origins are opened. So the DNA is unwound, everything is open, and the rest of the replication machinery is recruited, and replication then starts bidirectionally away from the origin. as I was saying before, lots of the studies are done in model eukaryotes, and this is a model based on findings in these organisms. And the thing is, because DNA replication is so central for cellular life, it was always assumed they would be the same in every other organism, right? Because there's an evolutionary pressure to keep it consistent and efficient as much as possible. So because you find the same in yeast and the same in humans, the claim of this is conserved across the karyotic tree has always been made. The thing is, now with the advances in science, in DNA sequencing, in metagenomics, etc., it has become clear that yes, this is a rough model of what is happening, but there are some variations across the eukaryotic tree. Some proteins are missing, or we can't identify them, or some others might be taking the function of canonical proteins. So what we know in kinetoplastids is that the machinery from the replicative fork, etc., it's very well conserved. So we can find all the players and everything seems to work as in model eukaryotes. All the machinery involved in the startup of opening the origin, etc., also seems to be there. The helicase is there. However, the loader is not. We can't find it. It's something that we're still working on, and we have a lead at the moment, but still has been 10 years trying to find that. So it's been not that straightforward and easy. It has a ORC complex, so the initiative factor is there, but it's slightly different, and that's what I'm going to be talking soon. And then the origins, it kind of is, but kind of isn't, like in other eukaryotes, and I'll also be brushing on that. So first, I'll focus on the initiation machinery and the helicase. So the model I showed earlier, it's very, very simplified. And here is just a tiny bit more complex one. So the initiator factor in eukaryotes is called the origin replication complex, or ORC. ORC is made up of 6 proteins, ORC1 to ORC6. And from now on, we'll forget about ORC6. We'll just focus on ORC1 to 5. It binds to the DNA as an open ring. And then another factor called CDC6 comes in, closes the ring around the DNA, and causes the ORC complex to change in conformation. That allows for the helicase to be recruited via the loader, which is called CDT1, and that is loaded onto the DNA. Then these 2 guys get out and then are called back again to load the second helicase, because as I was saying earlier, it's a bidirectional process. So you need 2 helicases, 1 on each side, to make up 2 replication forks going into opposite directions. So in kinetoplastids, has been a long journey to try and find the equivalence of all these factors. And here is just a summary of what we've been able to do in the last 15 years. So in 2005, and most of the things, all the things I'm going to talk about now are in studies all done in Trypanosoma brucei. We're also doing stuff in Leishmania, but it's quite preliminary, so I'm not showing that. So 15 years ago, the genome was sequenced, so that was very exciting. It opened up the whole field. And one thing that was very clear, as I was saying earlier, all the replicative machinery was there, replication fork was there, the MCM seemed to be there, so the replicative helicase seemed to be there. Couldn't find the loader, and then, surprisingly, couldn't find any of the ORC subunits except for ORC1. Later on, a group experimentally confirmed that ORC1 is ORC1. And more than that, that this ORC1 subunit also had a CDC6 function. And from there on, it was called ORC1 CDC6. And this was very interesting. And this was the reason why I started my PhD. It's because in archaea, there's only one ORC protein, and it's called ORC1 CDC6 because it has a dual function. So at the time when I started in 2011, the predominant idea or theory or hypothesis was that maybe trypanosomes are more like archaea doing the DNA replication than other eukaryotes. So are they the missing link? Are they not? So as a microbiologist, that was very exciting for me. And that was actually what sold me into the project. That didn't turn out to be the case. It was not as exciting as that, as finding a missing link. So later on, another group experimentally validated the helicase, showing that it is the replicative helicase, which was great. They couldn't find any of the other ORCs, but they found a subunit that was then called ORC1b because it was similar to ORC1, but also very similar to CDC6. And we are quite convinced, or we lean into the theory that it is a CDC6, although it's an odd acting CDC6 for other reasons. The person that was doing his PhD before me, so Calvin, he was able to find ORC4 and find other 3 proteins that might have been interacting with ORC1, but we couldn't find any link with ORCs. And that's what I did during my PhD, was trying to figure out whether they were part of the complex or not. Were they ORCs? Were they not? And turns out we finally got the link that they were actually ORC5 and ORC2. There's another protein that is not ORC3, but it might be doing this function. And that's what I've been trying to explore during my my postdoc as well, is that it is not. So this is how it stands at the moment. And there's a lot still to know. And I thought it was easier to guide you through this way rather than to show you all the data, because that was going to be really, really heavy. So what I'm just going to do now is just to guide you through what kind of techniques we use to get to these points. Instead of showing all the data, I'll just get one example and explain how we got to those kind of conclusions. So studying ORC in trypanosomes, we had an advantage. Well, there's an advantage in working with trypanosomes, is that you can use an inducible RNA interference system. RNAi is not so hot at the moment, now that CRISPR-Cas9 is in place. But it's a great tool, and it won a Nobel Prize 20 years ago. But in trypanosomes, it's a very useful tool. So what we can do is we can induce the cells to express double-stranded RNA with the sequence of our gene of interest. That will activate the RNAi machinery and will lead to a decrease in the mRNA levels of that gene. As a consequence, obviously, if the RNA is down, the protein will be down. And then we just ask, what's happening, right? And how do we ask this and how do we probe this? So the first question, which is the easiest one to answer, is, do the cells grow or do they not? If you interfere with DNA replication, you're expecting the cells to stop growing. So this is ORC1b, so the putative CDC6. And if you look at the cells that are not induced, so they're growing normally, they have normal levels of ORC1b, they grow happy. This is just passaging in the flask. But if you induce the RNAi, so if you decrease the mRNA and the protein levels of ORC1b, the cells stop growing and eventually start to die. So that was an indicative that was actually important for cellular growth, which was what we were expecting. How do we know it's involved in DNA replication? And is it involved also in cell cycle? So to do that, we can use, or we did use, microscopy. So the kinetoplastids, so like trypanosome, one cool thing about them is that because they have the nucleus and the kinetoplast, and their ratios change during the cell cycle, we can actually ask, is the cell cycle changing or not if you take that protein out? So here in the first bar is the normal profile, okay, without inducing anything. And then here, as you can see, even without reading what the legend means, the profile changes dramatically. And in this case, it changes to most of the population not having a nucleus, which is really dramatic and quite impressive. The other tool we used is to use an artificial thymidine nucleotides that can be detected by microscopy, and it's called EDU. Doing this assay is you add that to the cells, and then the cells that are replicating will incorporate it into their DNA, and then we can look down the microscope and say, haha, you replicated. And cells that are not replicating, obviously, are not going to put it into this DNA, so they're not going to be showing up. So they're not going to be bright. Then what we do is that we just count how many cells are, in this case, red. So again, without inducing, cells are replicating, are perfectly fine. And then you take out ORC1b and cells stop incorporating this analog. Besides microscopy, we can also do flow cytometry to ask exactly the same question. In this top graph is just DNA intensity in the nucleus. So you have cells in G1 and then cells in G2. And in between are cells that are replicating, right? Going from a DNA content of G1 and progressively into a DNA content of G2, which is fully replicated. Here in gray is normal cellular population. And here in the dashed line is what happens when you take ORC1b out. So as you can tell, straight away, it totally messes it up. And then here is the same assay as this one, but analyzing by flow cytometry. And here in this box is the cells that are replicating. And then when you induce, it's completely obliterated. So ORC1b is involved in DNA replication. So that was pretty cool. And we did this for all the ORC candidates. And that was what we saw. Not as dramatic as ORC1b, but same profile. What we can also do, and that's what we did, is to ask where in the cell are these proteins and do they change during the cell cycle? So if it is a protein involved in DNA replication, you're expecting it to be where? In the nucleus. So we wanted to make sure they were in the right cellular compartment, because that gives us an idea what they might be doing. So because we don't have antibodies against these proteins, what we did was to attach to them a very immunogenic tag, in this case, MYC, for which commercial antibodies exist. And then we just looked down the microscope. And here I'm just going to show the case of ORC1, and all the other ORCs mimic this profile. So here is the nucleus, there is the kinetoplast of one cell, that's another cell, etc. And here are cells in G1, S, G2, and here you already have two nucleus and two kinetoplasts, so it's post-mitotic cells. And as you can see, ORC1 is in the nucleus in every cell cycle. If we look at ORC1b, that is not the case. So some cells in G1 have it, all cells that are in S phase have it, and then some G2 cells have it as long as they haven't started mitosis. So with this assay, which was very simple, we could ask two questions. Where are they during the nucleus? Makes sense. Are they cell cycle regulated? Most of the ORCs are not, or at least in terms of localization and dynamics, they are not. We didn't ask, are they always bound to the DNA or not? That is something that we're going to do soon. But we were able to say they're there throughout the whole cell cycle. And there is one of them, which is ORC1b, that is actually dynamic. CDC6 is dynamic in other eukaryotes as well, but the time frame doesn't fit with ORC1b. So that's why I said it is a CDC6 candidate, but we're not completely sure it is actually CDC6 or other type of regulation. However, none of that answers the question of, are they in a complex, are they not? So for that, we did two techniques, so immunoprecipitations and also gel filtration, and I'll just very simply, I hope, explain what that is. So using the cell lines that I was talking about that we use for microscopy that have a MYC tag, what we can do is burst the cells out, purify their proteins, and then incubate them with magnetic beads that have antibodies against MYC. We incubate them, so what's going to happen is that they're going to grab, let's say, ORC1, And then when we cleave that connection between the antibody and the MYC, ORC1 and everything that was interacting with it, hopefully in a complex, will come out. And then we send that for mass spectrometry, and we identify what proteins there were. So that's how Calvin, for instance, and I think you can't see the references because I didn't make the window big enough. Sorry about that. That's exactly what Calvin did, which was the PhD student before me. And that's how he identified the putative candidates for other orcs. What he also did, and also what I did later on, was to have, for instance, a cell line that has ORC1 with the tag MYC, and pick up another ORC candidate, in this case, what I'm going to show is ORC4, and tag it with another tag, in this case, HA, and then use antibodies against HA to pull them down. And then ask by Western blot, which is a method to detect proteins, whether they come down together. And the answer was yes. So here was ORC4, and he pulled out ORC1. This only tells us that they interact with each other. It doesn't tell us, are they in a complex? So to try and answer the question, is there an OR complex? What we did was gel filtration, which is to pass the cell's protein extract through a matrix. And what it does is that complexes of different sizes come out at different times. So in this case, larger ones come down first, medium, and then smaller ones. And then we just run it on a Western blot. And that's what we did. We ran a cell line that had ORC1 and ORC4 tagged so we could detect them. And in parallel, one that had a subunit of the helicase. And what we could see was that there were some fractions where the three of them appeared, and those fractions corresponded to sizes that corresponded to the expected size of an ORC complex connected to a helicase, for instance, or just an ORC complex, or just an ORC with a helicase, with one helicase or with two helicases. So that's how we came to the conclusion that yes, they are eluting in a complex of the same size, and we think it's the same complex. So that's how we got to this, where there is an ORC, but it's not so straightforward. It took a lot of bioinformatical effort as well to figure out that they were ORCs, but it wasn't as clear cut as picking up, for instance, the yeast proteins and ask, are these ORCs? Because they'll find that in the databases. So the sequences are very, very different. So even ORC2 and ORC5, the sequences are incredibly different. That it's very hard to pick them up in bioinformatics searches. The other thing I wanted to talk about, and that's so just as a conclusion, sorry, there is an ORC, but it's very divergent, which was what I said. And then there's still lots of questions that we want to answer. One of them is, how is the ORC organized? So are the different subunits placed in the same conformation as, for instance, yeast or Drosophila? Or are they differently, or are there other players? How are they regulated, especially ORC1b? How does that come into place? And does that activate the ORC complex? Does it activate the helicase? What is going on? Are there any other layers of regulation? And then, I don't think you can see it, if there is a loader. And that's what we've been exploring in the last year. The other thing, and that's the last thing I'm going to talk about, and also I'm running out of time, is origins, right? So we tried to map origins of replication in both trypanosomes and Leishmania. And to do that, we used a technique in which we pick up cells that are in S phase, so they are replicating, and cells that are in G2 phase, which have completely replicated their genome. We pick them up and we sequence their DNA. And then we do what we call marker frequency analysis, or MFA-seq, which in reality is just to divide the reads in S phase over the reads in G2. What that means is, let's say chromosome 8, in the sample from S phase cells, these cells have just started replicating or are replicating. So what you're going to have is DNA sequencing reads. They're going to align to the chromosome. And some regions haven't started replicating. So there are some reads there. But then there are other regions where the replication fork already started. So there are regions that already have extra copies, right? Because they are being replicated. So you see an accumulation of reads in those areas because there are extra copies. So there's extra DNA to sequence. So you have more. In G2 cells, they already replicated everything, right? So the reads will be more evenly distributed across the chromosome. What also happens is that we know that when you do DNA sequencing, the sequencing is not completely flat or even throughout the whole chromosome. There are regions that are sequenced better than others. So to get rid of that bias and that technical issue, we normalize the S phase sample over the G2. And in that case, you get rid of artifacts, but you keep the regions that were already replicating. And then you just plot in something like this, and the peaks should overlap with origins, or there are regions that are being replicating. So either origins or origin-rich regions. So we did that for both organisms, and T. brucei has 11 chromosomes, 11 megabase chromosomes. There are other types of chromosomes, but they're not the core genome. This is the core genome, so it's 11 chromosomes. I don't think you can see the 11th, but it's there. Leishmania has 36 chromosomes, but to make it easier, I just plotted 11 for comparison. What you can see in brucei is that it resembles more or less what you see in model eukaryotes. So you see several peaks per chromosome, and the number of peaks actually increases with chromosome size, which is what you would expect. We also have data for where ORC binds, and that overlaps with the peaks. And there are more ORC binding sites than origin peaks. So that all goes together with the present model of DNA replication. Leishmania is a bit not there. It's very odd. If we do exactly the same technique, you have one main peak, and then it's either very noisy or very flat. So it seems like there's a strong origin and then not really much. The problem here is that this is the first eukaryote where you can detect one origin, which was pretty cool at the time, but it's not enough to completely duplicate the chromosomes within S phase. So although we could only detect one clear origin per chromosome, we always knew that they need to be doing something else, because otherwise they don't have time. So that is one thing that we've been working a lot, is to try and understand how Leishmania replicates. How do they have one main strong origin? Do they have others? The problem is this technique is that is a population-based. What that means is we'll be able to detect origins that are activated by the majority of cells. If origins are only activated by a couple of cells or a small population, they'll be lost in noise. So what we've been doing in the recent years is to try and adapt other more sensitive techniques, such as trying to do this in single cells, which has been a bit hard, but it's going. And then the other thing is to sequence a group of cells by using a methodology that actually sequence one big molecule at the time. And we can detect if there's one origin more or where several origins are. So we hope that we're going to get a clear picture soon. We don't know exactly when. But that's a big focus of research in the group at the moment. And it's still a big puzzle, to be very honest, but it's really interesting. So they're very different from model or other eukaryotes, but Leishmanian trypanosomes are very closely related, relatively closely related. But still, they're very different in the way that they replicate their genome, although all the proteins are there. So all the ORCs that I mentioned in brucei that we studied, we can find them in Leishmania, and we've been studying them as well. So they're there, but things are happening in a different way. So this is just a final summary. The message I want you to take home is that from the moment the DNA opens, everything seems to be there and very similar to what other eukaryotes do, at least the ones that we've studied that we know. The helicase is there, but the loader isn't. And that in itself is really interesting. Also because archaea don't have a loader. So ORC is there, but it's very different. And then the origins, especially in Leishmania, there's something there that is strange, and we just need to explore that further. The advantage or the potential advantage of this stage being different in these parasites is if that it is significantly different from the human steps, we can then use that as potential drug targets to tackle the diseases. So it's interesting in several ways, not just because of the biology, but also in terms of human health and trying to tackle these pathogens. So that's me. This is our lab. So we are the McCulloch lab. Here is the boss. This is the last pre-pandemic picture we have. We've changed a bit, but lots of us are still there. These are the names of lots of people that have been involved over the years, trying to decipher how these guys replicate. These projects have been funded by the Wellcome Trust, the BBSRC, FCT during my PhD. All the diagrams I've showed you today, I actually did it in BioRender, which is an open source website where you can get lots of biology and scientific and medical illustrations. So that's pretty cool. Check it out. And we are based at the Wellcome Trust Center for Integrative Parasitology. Here's the Twitter handle, the website, and then if you could see, but you can navigate to the website, we have lots of comic books, and that's where I took that picture from. Lots of comic books about the parasites that we study. We don't have for Leishmania, but we have a comic book for trypanosomes, malaria, helminths, and toxoplasma. So go and check it out. It's a good public engagement tool. And then I'm on Discord with this handle. I joined BioCord a few weeks ago. This is my Twitter handle and the group's handle as well. And you can also find me on LinkedIn. And I hope you enjoyed it. I'm sorry about the technical issues. I was really not seeing that happening. I hope it wasn't too technical or too confusing. Anything, just get in contact. And that's it. If anyone is looking for PhDs, etc., go and check out the PhD programs at the uni and even the institute. And if you're a postdoc or you're wanting to do a postdoc, just contact the PIs. They're more than happy to reply to you, even if they don't have funding at the moment, to help you apply for funding to come. So I think that's it. And thank you very much to BioCord for the opportunity. And
Thank you for coming on. So I think we have a couple questions here and then one question on Discord. So I'll just read out the first. Yeah, I'll read out the first one. It is from a clinical point of view, what can be done in the treatment and prevention of Trypanosomas species? Most of the methods I have seen rely on vector control.
Yes, so I think lots of it is, you're completely right, is on vector control. There's a lot of research on developing new anti-trypanosome drugs, but it's quite hard. It's also it has been quite hard for a while in the sense of, because it's not a major disease. Actually, trypanosomes and Leishmania are considered the neglected tropical diseases because there's not that much interest from the pharmaceuticals to develop drugs against these, because they don't affect that many people. But what has happened is that several years ago, I don't remember how long ago, but there was a big consortium, and I think it was pushed up by the Bill and Melinda Gates Foundation to join the pharmaceuticals and get them to offer drug libraries to academia, so people can test thousands, millions of drugs that they have in their libraries, against these parasites. They develop them for cancer or for other diseases, but they just make them available to academic groups to test them against these parasites as well, because if they work, they've already been developed, which is great. They're already commercial, they already passed all the FDA, etc. approvals. And also, if they don't work completely, at least you can start modifying the skeleton of the drug and trying to modify it. So when I worked in Dundee, there's a whole building that does drug discovery on neglected tropical parasites and malaria and tuberculosis. And they get lots of those collaborations with pharma. Yeah.
Uh-huh. I think that was the answer to that question. There's one more question on the research on ox. So can the research on ox help in terms of curbing the antigenic variation these organisms present to avoid the immune system?
So that is a very good question. That was actually the title of my PhD when I started, and then I kind of ditched the antigenic variation bit because there was so much going on the replication side. So there are one paper or two papers where they've seen that if you do RNAi on ORC1, you start seeing deregulation of the VSG, which is the surface coat. The thing is, my issue with studying is not studying, but going too much into the antigenic variation bit while studying ORCs is that if you take out ORC, you mess up DNA replication, you mess up nuclear structure and genome structure. And what we know from antigenic variation is that the expression of a single surface coat protein is very related to nuclear structure and placement of the transcription sites and all the machinery. So if you take down ORC, it collapses all of that. So it's like chicken and the egg kind of question. Is like you take ORC, there's antigenic variation, but was it really ORC that was acting on it, silencing the other sites, etc., or is it just that everything is collapsing, and then as a side effect, you get antigenic variation? However, I think it's worth looking at it specifically because ORC1 CDC6 is also thought to be involved in silencing of the subtelomeric regions. So yeah, so I think it's worth taking a look, although I'm more interested in the DNA replication bit, but definitely it's something that is worth taking a look and explore a bit more. Yeah, that was good.
That was a good question. So yeah, there's one more question on Discord, which is, are the ORC analogs in humans similar to the ones which are seen in trypanosomes, or are they significantly different from the trypanosomal versions? And could this also be a target for therapeutics if they are different enough?
Yes, definitely. So that's what I was trying to say is that the human ones are known. There's actually the structure for the human ORC, which is pretty cool. And ORC1 CDC6 is similar. So it's similar. We can tell, like if you align the ORC1 CDC6 from trypanosomes and the one from humans, it aligns and you can see, conservation of certain parts. However, the human one has an extra domain that the trypanosomes one don't. So although the trypanosome one is quite conserved, it lacks a huge N-terminal domain, which is involved in silencing, actually. ORC4, it can also align, but it's quite hard. And then ORC2 and ORC5, if you use, for instance, BLAST, it doesn't pick up the human one. So that is one of the things, that was one of the hypotheses, it is one of the things we've been working on, is are they sufficiently different to use them as drug targets? Yes, so that was a great question, and that is one of the aims. So thank you very much. Yeah,
I think that's the end of the questions, but I think there was some requests on the server for you to repeat your intro because they could not see the initial slides.
Oh, if you want me to do, I can do it really quick.
Sure.
Are we running? Okay.
This time.
Oh, that's time. Okay. Right. Okay. Sorry, going really fast. Or maybe it's easier to just get out of it. Yeah, so I think this is where you lost me. last time. Yeah. So, hello, this is me. So I'm Kat. I'm a microbiologist and come from Portugal. She's there. And I did my undergrad in Lisbon and also my master's. And I did it in microbiology, and I was really interested in bacteria and viruses. I was not really into parasites, but in my final year, I had the awesome opportunity to work in one of the great malaria labs. And then I was hooked ever since, and I never left parasitology. I wanted to do a PhD, and I wanted to do it somewhere else for the experience of living in another country, learning another lab culture, learning another culture. So I moved to Scotland. It was not that far, but it was different. And I did my PhD there, and that's when I started working DNA replication in trypanosomes. And then halfway through, I kind of picked up Leishmania as a side project for the origins that I showed you at the end. I then moved a bit north around here to Dundee, where I worked on antigenic variation, like the expression of the surface protein and also on cell cycle regulators in trypanosomes. And I really wanted to go back to DNA replication, so I came back to my PhD lab, and that's where I've been exploring new avenues that I uncovered during my PhD, and that's what I've been doing, and that's what drives me. So that's it. I'm really sorry you did miss that the first
That's all
time.
right.
I had my cartoon and everything, but like,
Yeah. I think we wanted to see the cartoon really bad.
yeah, so that was it. Sorry. Yeah, I hope
Yeah,
you enjoyed. And thank you
I
very
did.
much
I
for the
did enjoy it.
opportunity.
Yeah.
Okay, thank
I
you.
really enjoyed it. Yeah. Thank
Yeah.
you. Let's.
And-
I hope to see you around on the server more. Let's plan more events.
Yeah, just let me know. And if you guys
Yeah.
have parasitology questions that I can answer, just drop me a line
Definitely.
on Discord and
Mhm.
I'll follow through. Okay.
Yep.
Thank you very
Thank
much.
you very
Have a
much.
nice day, guys. Bye-bye.
Yep. Thanks. Bye-bye.
Bye.